Thanks very much for joining the Alberta Transplant Institute rounds. Today we're going to be talking the second part of our presentation on molecular rejection states. So before we start, we have to do the land acknowledgement. We acknowledge that the University of Alberta is located on Treaty 6 territory and respects the histories, language, and cultures of our First Nations, whose presence continues to enrich our vibrant community. We're joined today by Pat Gauthier, who's a senior data scientist at TSI. He's a PhD in environmental toxicology and postdoctoral training in Calgary and at the U of A. His earlier work has focused on drug and pollutant interactions in aquatic models, fish. But he comes to us with a remarkable knowledge of how to handle big data. How to handle high dimensionality data in particular, and now he's focused on trying to improve the diagnosis of rejection. And we'll also be joined by Katelynn Madill-Thomsen, a research scientist with a focus on the application of gene expression. She did her PhD with us and is currently working as a senior research scientist for Transcriptome Sciences, Inc., and working on all the organ transplants. So without further ado, I will then go to our presentations, and I'll share screen. And so what we're trying to do here is assess rejection states at a molecular level. And today we'll be focusing mostly on transbronchial biopsies, mucosal biopsies from lung transplants, and also on liver biopsies. These are my disclosures. Sorry. Let me go back and just do that again. And this is the team that works on the MMDX side. It's quite a team of talents, and we're very grateful to our support from Natera, from Thermo Fisher Licensing Agreement, originally from the Genome Canada project.
Well, we're not seeing your slides, Phil.
Okay. Go back. Okay, now you see?
Yep.
Okay, good. So our learning objectives today are that we'll update the new data on the changes associated with T-cell mediated rejection. We're seeing this in all the organs, and then we'll be focusing on the lung and liver. Let me take you back to 1912 to the Nobel laureate lecture by Alexis Carrel. Dr. Carrel was a young physician in France who was very impressed by the fact that when the President of France was stabbed and an artery was cut, he bled to death because no one could fix arteries. So he went to a seamstress and learned how to sew arteries. And then he had the idea that if he could sew arteries, he could sew in an organ transplant. He started to sew kidney transplants in dogs and cats. And by histology, he said that there was a diffuse nephritis, an inflammation that occurred in dogs and cats after about 5 days. Of course, these were allogenic. These are between individual dogs and cats. He said it's very probable that a reaction of the organism against the new organ takes place after a few days. So this is the first description of T-cell mediated rejection in organ transplant. And it's actually been the target of many of our activities. Is the driver of all immunosuppression. It was the endpoint for all previous drug trials. And now for the first time, I think we're able to take apart what the inflammatory infiltrate is in the graft and look at the parts. Now, this is part of what I think is a new beginning, where we're on the cusp of a new era that's going to change the outcomes for the first time in 20 years in transplants. And this activity will require the support of new laboratory tests for diagnosis. You need to diagnose, you need to see the effect of treatment, and you need to see relapse. So MMDX is a system now for looking at biopsies, and it's been developed in 7 large-scale clinicaltrials.gov programs. We'd like to thank our collaborators for their past and ongoing support, especially the Natera support recently. We'd like to thank the thousands of patients who've consented to these studies, and we wouldn't be here without the consent of these many thousands of patients. we are the mothership for a series of licensed laboratories now in the United States, Czech Republic, Austria, and Brazil, and others are in progress. And this gives us access to a very large number of biopsies, which we can use to increase the power of the studies. So MDX has now analyzed biopsies, and I'll be showing you the results in microarrays and walk data sets for 10,500 organ transplants, including kidneys, hearts, lungs, and livers. And you know some of the ways that we characterize these. We take each biopsy and look at its rejectionness using classifiers. And this is looking at its molecules trying to establish what the rejection states are in every biopsy. Then we distribute the biopsies in principal component analysis. Principal component 1 being all rejection, principal component 2 being antibody-mediated rejection, being T-cell-mediated rejection. And then in kidneys, principal component 3 are the stages of antibody-mediated rejection: early stage, fully developed, and late stage. We can also represent this, and you see the reports. We represented it with the UMAP, so we can look at it in 2 dimensions. And we can do this in one way or another for all the organs. But now what we can do is something different. We can take the output from that. So each of these 10,000 organ transplants we have 500,000 measurements of every probe on the microarray, which is a very large amount of data. So we can now take this data, and in each organ we can look at the signature matrices. We're particularly focused now on this one described in Nature Medicine in 2015 called LM22 CyberSort. We'll be showing you the results from that, but there's a large amount of data now is accumulated about how to estimate cell types in high dimensionality data. Now, these are the cell types I'll be telling you about, and they're each given abbreviations. It's hard to give a presentation on 22 different players, but these are the cells we'll be talking about. I'm sorry we have to use the abbreviations because it's just otherwise too much text. We're talking about naive and memory B cells, plasma cells, CD8 T cells, various types of CD4 cells, including CD4 naive, CD4 memory resting, CD4 memory stimulated, T follicular helper cells, and regulatory T cells. We'll also look at gamma delta T cells and resting and stimulated NK cells. And the myeloid cells, we'll be looking at monocytes, macrophages day 0 unstimulated, macrophage classically activated, macrophages alternatively activated. Then we're looking at dendritic cells, resting and dendritic cells stimulated, mast cells resting and mast cells stimulated, eosinophils and polymorphonuclear leukocytes. And this is just some of the background showing there's a very large amount of data and referencing to define these cell types in the Nature Medicine paper in 2015. And we can give you more details on that as you. So what we're seeing in MMDX, when we take MMDX and analyze it by CyberSort, we see that T-cell mediated rejection is identified in all the organs, but antibody-mediated rejection, as defined as a molecular state in kidneys and hearts, doesn't seem to exist in that state in lungs and in livers. The molecular files generated were analyzed to estimate inflammatory cell populations. This method is also called cellular deconvolution, using LM22 and kidney transplants. First of all, the kidneys with T-cell mediated rejection, MMDX diagnosed, had a 4.5-fold increase in inflammatory cell burden compared to those with no rejection. And then there was a change in the percentage of the cells. And when you combine the percent change with the fold change in total burden, you see the most selective feature of T-cell mediated rejection was a 172-fold increase in memory stimulated CD4 cells. They still constitute only 1% of the population of the cells in T-cell mediated rejection, but they're remarkably changed. This is accompanied by an increase in other cell types, gamma delta, CD4 follicular helper, and CD8. Also in classically activated macrophage. Now this is the effect of gamma interferon. So the one universal effect aspect of all rejection is an increase in gamma interferon inducible genes. And in our case, we're looking at classically activated macrophages receiving gamma interferon in the context of other cytokines. We also see an increase in memory B cells and plasma cells and an increase in eosinophils. And perhaps in stimulated mast cells as well. So that's T cell mediated rejection. There was a decreased percent of regulatory T cells, but the actual number didn't change. So that it wasn't that there was a decrease, it's just that they didn't increase. T cell mediated rejection in heart, lung, and liver looked almost the same. A remarkable conservation of this complicated cell play that we're seeing. The results suggest a central role for a population of CD4 memory stimulated T cells in the cognate recognition event that actually organizes the entire transplant process in T cell mediated rejection, possibly engaging a memory B cells antigen presenting cell. And I'll just show you those results. So in each of these, we have looked at the biopsies that had no rejection, possible T cell mediated rejection, mixed, possible antibody mediated rejection, antibody mediated rejection. So just look mainly at the gray, the red, and the blue. So the most remarkable change is this big change from almost non-existent to significantly increased, 174 increase actually in the T cell, in the CD4 memory stimulated T cells, accompanied by increases in the other T cells, CD4, T follicular helper, CD8, and gamma delta, accompanied by an increase in the gamma interferon affected macrophages, M1 classically activated, and an increase in memory B cells and plasma cells, suggesting that they may be, the memory B cell may be the antigen presenting cell in T cell mediated rejection, which would be compatible with other literature on how T cell inflammatory processes work in a long-term state. There's also an increase in monocytes, activated mast cells, and eosinophils. There is not a significant change in the dendritic cells, which is something we were expecting. I mean, we thought we'd see more of a change in the dendritic cells, but the fact is it may be the memory B cell is the story in terms of antigen presentation. In antibody-mediated rejection, we see more subtle changes, as you'd expect. So we see a mild increase in stimulated NK cells. But the odd thing is that the CyberSource says there's a lot of stimulated NK cells in all organ transplants, whether or not they have rejection. And that's something we come back to. But there is an increase in stimulated NK cells in ABMR, and it's the most remarkable feature. There's also an increase in gamma interferon affected macrophages, the M1CAs. And there's an increase in stimulated mast cells. So they may be part of antibody-mediated rejection. There's some margination probably of monocytes as well. And the Treg percent is decreased, but the absolute number is actually preserved. It's just that they aren't increased. So there are some decreases as well. The mast cells resting may be transformed into mast cells stimulated. And what this suggests to me is it takes me back to the days when in medical school I played football with the meds team. We would play the other faculties. We could always beat arts. We could never beat engineering. But I remember my job as a linebacker was to sack the quarterback. Now in T cell mediated rejection, I think the quarterback looks like the cognate signal 1, signal 2 engaged memory T cell. If that's true, we could now target T cell mediated rejection, which remains a serious and not well controlled disease in organ transplants and causes many organ transplants to be lost. So with that thought and memories of my misspent youth trying to play football for meds, I think I'll turn you over to Pat, who will take you into lungs. Pat, could you share your screen?
Yeah, thank you. I am getting a notification here. It says cannot start screen share while other participant is sharing. Okay, I have bypassed that. Can you see my slides?
Yes, we're good.
Okay. So I'll go over the current state of molecular diagnostics of rejection in lung transplants. And there are a couple of diagnostic tests that are available, and there's some fundamental similarities regardless of which test you choose. And the idea is that diagnoses of rejection are based on gene expression profiles quantified in a biopsy. And you compare that gene expression profile to validated profiles that have been developed in a large population of biopsies containing a molecular rejection model. And one step, of course, is to use marker genes that are selective for no rejection versus all rejection, but also selective for ABMR and TCMR to distinguish these rejection states. But we've learned that gene expression profiles for ABMR and LUNs are complicated. The hallmark genes of ABMR in kidney and heart that we've used don't adequately separate TCMR from ABMR and LUNs. And so this molecular rejection state remains elusive. But really the goal in separating these two states is to provide targeted therapies based on known cellular involvement in the state. So one option, given the complexity of the gene expression profiles of ABMR and TCMR, one option is to bypass that and to directly measure the cellular involvement. So cellular resolution and diagnostic testing could help resolve complications in distinguishing ABMR from TCMR. So I'll go through the steps that we use for building a molecular model of rejection. And step 0 is really defining a target. So what do we want to identify? And simply put, we could include no rejection, T-cell mediated rejection, and ABMR, antibody mediated rejection, as our targets. And with those targets in mind, then we do feature selection. So what features do we use or include in our model, which are going to help us identify and characterize those targets? And for a purely molecular rejection model, these can include individual gene expression data, multi-gene scores, classifier probabilities that were derived from gene expression data. And there's also a question of, is there an optimal selection of features? So we could actually do a genome-wide model, including every gene in the human genome. But we know that this isn't going to produce as accurate a model as one that only includes a truncated list of features which are truly selective for the targets that we're interested in. And then one other step, which is fairly ubiquitous in developing these models, is feature transformation or dimensionality reduction. So in Phil's slides, he's already presented principal components. So given the sheer number of features that can be included here, it can be quite complicated to interpret them and the variation associated with them. But what principal component analysis does is compresses all that variation into vectors which are more easy to present. Step 2 would be, what type of model do we want to use? Are we going to use a supervised model or an unsupervised model? Well, supervised learning is challenging when the histologic definition is under development and the gold standard is elusive. So for LUN, what we've opted for is unsupervised models. The caveat being is that an unsupervised algorithm is going to give you clusters, it'll give you labels, but it doesn't tell you what those labels represent. So careful interpretation has to be done post hoc to annotate those labels. And in order to do that, what we do is we let the data guide us. So this can be done using individual gene expression data or multi-gene scores within the biopsies. We could also look at clinical features of the patient at the time of biopsy as well to help us annotate these labels. And if follow-up data are available, we can, of course, do risk stratification based on the label, but we could use the risk stratification as a way to validate the annotation. So for particularly dangerous disease states like TCMR, we can use follow-up data to validate the label annotation. And then the final step would be actually producing the diagnostic report. And so it's a big leap from step 3 to step 4 here. But really, after you develop the model, the question is, how do we use it and incorporate it into a diagnostic test? And for the MMDX, it's based on expert signouts from a myriad of molecular scores for each biopsy. So we're not just using the label assignment that's provided by the clustering algorithm. And the reason we don't do this is because we're measuring things on a continuous scale with gene expression data. And so a continuum from no rejection to rejection, or from ABMR to TCMR. And by simply using label assignment, we don't capture that granularity. And a final piece for our diagnostic reporting that we're moving toward is automated signouts. And the reason being is that it's going to greatly increase the diagnostic throughput of these tests. So a bit of a segue coming back to digital cytometry and CyberSort. As I mentioned, directly measuring the cellular composition could be a means to overcome the complexities of mapping the gene expression profiles for ABMR and TCMR in lung. But I predict that during any of our lifetimes, we're not going to see single-cell transcriptomics used in a diagnostic platform for transplant rejection. The analyses are very laborious, very low throughput. A lot of work has to be done to ensure consistency across assessments in a diagnostic setting. So it seems like quite a challenge to incorporate that cellular resolution into diagnostic reporting. But there's been a large consortium of international researchers who have been on a quest to get robust cellular enumeration from bulk transcriptomics data. And so the idea is that you have your leukocyte infiltrate in the tissue of interest. You completely bypass the single cell analysis, instead opting for bulk transcriptomics on a platform that is higher throughput and has better consistency across samples. And then from that bulk transcriptomic data, we do our cellular estimation based on the gene expression profiles to get our cell composition estimates. And a major benefit of this endeavor is that it allows for assessment of large, already collected databases. And these databases can represent millions to tens of millions of dollars worth of investment that's already been made. So the opportunity here for really increasing the data value that we can get out of these populations through digital cytometry is really, really attractive. Okay, so now getting to Interlung. So Interlung is a large multinational study involving 19 investigators from 9 centers. And it provides us with a unique opportunity to evolve our understanding of the molecular landscape of rejection in lung transplants. One really interesting feature of this that I think I need to emphasize is that we use exclusively microarray technology, and there's a big benefit to us using it. One is the high manufacturing standards of microarray chips and the normalization of the gene expression data that we get from those chips allows us to integrate a evolving process of developing molecular rejection models. So maybe the first model that we develop uses 100 biopsies. The second uses 200, but that 200 includes those first 100. So the contributions from these centers and these patients are just continually being used to develop larger and larger models, hopefully coming to a closer and closer approximation of the truth for transplant rejection. So here's a workflow for Interlung that we're moving toward. We have 2 different types of biopsies. We have the transbronchial biopsy, but we also have a third bifurcation mucosal biopsy. And so the cohorts for TBBs and 3BMBs are quite large, but we also have a paired TBB and 3BMB cohort, which is really interesting to us because the TBB is a relatively dangerous procedure and actually excludes some patients from being biopsied altogether. So our question is, with a 3BMB, can it act as a surrogate, or can it augment the TBB, or eventually could it even replace the TBB in terms of molecular assessments of rejection in lung transplants? So once we have our biopsies, we do the microarray transcriptomics for bulk transcriptomics. We get the gene expression profiles for the biopsies. We use that profile to calculate transcript set scores, as well as carry out cellular deconvolution to estimate the cellular composition for each biopsy. And then after this is when we start to develop our molecular rejection models. So we use unsupervised techniques. Once we get the cluster assignments, then we start to annotate those clusters. We do this with gene expression profiles, pathway enrichment profiles, and now we're actually including cellular profiles in this annotation as well. And once the groups have been annotated, then we can look at graft outcome and risk stratification for the model. So for Interlung TBB, major question that we asked is, can molecular features distinguish ABMR from TCMR in TBBs? And so the figure on the right here is a principal component space of biopsy set from our previous model. On the x-axis PC1, on the y-axis PC2. Each dot represents an individual biopsy, and the large circles represent the cluster location. So in this case, these are archetypes. And so this model was derived using a relatively non-selective set of rejection-associated transcript sets. And we were able to isolate a group which was consistent with a TCMR profile, but not ABMR. We couldn't get ABMR from this analysis. And one question that we had is, we're relatively successful at doing this, separating ABMR from TCMR in kidneys and hearts. So what knowledge can we glean from those assessments to help guide us improve our TBB models? So principal component 2 in kidney and heart is really a major canonical vector separating ABMR in the positive PC2 space and TCMR in the negative PC2 space. So a question that we had is, can we retrain this model with this in mind to try and reorganize these biopsies to mirror the separation of ABMR and TCMR and PC2. So we came back to the drawing board and included a much truncated list of features, which were more selective for the labels that we were interested in. And we did use a certain optimization for feature selection here. I don't have time to get into it now, but maybe if there's questions, certainly we can get to it at the end of the presentation. But this is the model which we ended up with. So on the left-hand side, again, the biopsies projected in principal component space, PC1 on the x-axis, PC2 on the y-axis. So in terms of annotating these labels, we identified 2 no rejection states. One labeled NR and the other labeled NR low SFT. So this biopsy set had really low surfactant gene expression. And the way that we annotated these was by looking at a variety of different molecular scores and individual gene expression data and comparing them to the other groups. So CD45 is a pan-leukocyte marker indicative of leukocyte infiltrate, and it was lowest in the no rejection groups. Gamma interferon transcript set was lowest in those groups as well. And really, across the board, all the rejection scores were lowest for the no rejection groups. We isolated a TCMR set in red here. And the reason why we label it as TCMR first was that it had high CD45 and gamma interferon, but it was elevated in our TCMR gene set, T cell burden gene set, a macrophage gene set, as well as individual genes, CTLA4, which is a T cell marker, and ADAMDEC1. So this was convincingly TCMR. Then we ended up with this blue cluster up top, which was would be the putative ABMR cluster that we were searching for. But we labeled it NKRL, which stands for NK cell-rich rejection-like state. We knew it was a rejection-like state because it was elevated in CD45 and gamma interferon. It also had elevated expression of granulysin, which is it's associated with ABMR, but perhaps not really selective for ABMR. It had higher NK cell burden and higher DSA selective transcript sets. But it was quite a large population, 244 biopsies, and we knew that from prior literature that ABMR is a relatively rare state, and so it seemed unusual that it was such a large cluster. So pushing us away from identifying it as ABMR. When we looked at clinical associations with these groups, actually NKRL was biopsied less for indication than no rejection, had less dysfunction than no rejection, and less of an association with CLAD than no rejection. And those are 3 key markers that have been identified as being related to ABMR in LUNS. So again, pushing us away from thinking the state was antibody-mediated rejection. In terms of outcome, for NKRL, it had the same incidence of graft loss as no rejection, whereas our TCMR group had an elevated incidence of graft loss. We then looked at the cellular profiles within these cluster assignments. And we looked at the selectivity for TCMR first, which we were convinced had represented a TCMR-like state in transbronchial biopsies. And the features that were most selective for TCMR were the CD4 memory stimulated T cell, CD4 T follicular helper cell, M1 macrophages, and CD8 T cells. So as well as plasma cells too. So again, pushing us toward being confident that this cluster truly did represent TCMR. And actually, if we took this cellular profile and used it to predict TCMR within our biopsy set, it predicted TCMR with really high accuracy, so 0.95. we did the same approach for NKRL to see if we could glean any more from this biopsy set. And interestingly, the resting NK cells were the most selective feature for NKRL, followed by monocytes, neutrophils, and stimulated mast cells. But what was absent here was an elevation of stimulated NK cells. So again, we weren't necessarily convinced this was ABMR because we would expect stimulated NK cells to be elevated. Okay, so that was TBBs. And then we had the same question for 3BMBs. Again, can we distinguish ABMR from TCMR in 3BMBs? So here's the previous model that we had developed. Again, using a non-selective set of rejection-associated transcript sets. In red here, a rejection cluster was identified, but it wasn't confidently estimated to be TCMR or ABMR. And so we had the same questions. What can we glean from our knowledge of kidney and heart and now TBB in terms of guiding a new model to isolate TCMR from ABMR in mucosal biopsies? So we used a truncated feature. Many of these overlap with what I showed you for TBBs. So again, molecular scores that were selective for rejection, TCMR, and ABMR. And here's the model which we ended up producing. So on the left of this plot, the biopsies are projected in principal component space. And we identified a no rejection state, just as we did for TBBs, by observing that this state had the lowest scores for rejection compared to all other biopsy groups. We had two clusters which seemed to be indicative of TCMR, particularly TCMR1, which was quite elevated in CD45 and gamma interferon, as well as TCMR, gene set score, T cell burden score, macrophages, CTLA4, and ADAMDEC1. Similar elevations were seen in TCMR2, but to a lesser extent than TCMR1. And then for PC2, we ended up with this group that we've just called other rejection for now. This hasn't been published yet, so this name may change once we finalize the paper. But we knew this was a rejection-like state because it had elevated CD45 expression as well as gamma interferon effects. It did have higher granulysin. It had a higher NK cell burden, and the DSA selective transcript sets were higher in this group. But when we looked at clinical information, we saw that, first of all, TCMR within this model had higher, more likely to have been biopsied for indication, higher dysfunction at the time of biopsy, and had association with CLAD and DSA. But the same, we couldn't say the same for this other group. So pushing us away from labeling this as an antibody-mediated rejection group. Also, within 3 years, there were no failures within this other group. Whereas for TCMR, particularly in the first and second year, the incidence of graft loss was higher. When we looked at selectivity of the cellular profile, we saw very similar pictures we saw with TBBs. So CD4 memory stimulated cells, M1 macrophages, gamma delta T cells, and CD8 T cells. And when we looked at it for the associations were quite weak. And the highest features that were positively selected for other one were actually monocytes and neutrophils. Okay, so then we were curious, how do these two models compare for TBBs and 3BBs? Are they going to give you the same answer? Can they be used interchangeably? So what we looked at first was correlations between key rejection scores and key TCMR scores within the TBB and 3BMB, and found fairly good correlations, modest correlations with these scores. So the molecular information was being shared to a certain degree. We then looked at overlap in the prognostic value of these two models. So if we looked at diagnoses by no rejection in 3BMB and TBMB or both, we found no significant difference in the prognostic value of the model, which means they can be used interchangeably in that regard. For TCMR, we did the same thing and found that there was no difference in the prognostic value of either the TBB or 3BMB model. Okay, so just to wrap up, we've mapped a molecular TCMR state in lungs, but not ABMR, and some questions as to why we're struggling so much. One is does size matter? So the lung is quite large, much larger than the kidney and heart, and the large microcirculation of the lung, it challenges our current model for ABMR processes, which were based on kidney and heart. So these processes may indeed be going on, but because the large microcirculation, it's possible they're just not reaching a critical mass where injury is being induced through the ABMR process. One other question is too soon. So most of these biopsies were sampled 1 year post-transplant, and we know that ABMR is very slow to progress. NK cells as well. So the lung has tissue-resident NK cells, and this might complicate ABMR-related gene cell expression signals that we're getting in these biopsies related to ABMR. And so ultimately, cellular resolution may be essential to help us resolve this complication. Okay, so that's all I have. So I'll pass it over to Katelynn.
Katelynn, could you share your screen now?
Yeah, I just have to, it says I have to wait for a second.
If just one moment, Katelynn, before you start, are there any immediate questions from some of you lung people for Pat about his representation of the value of the mucosal biopsy? If not, we'll go on to liver.
Okay, so thanks, Phil and Pat. There's going to be a lot of crossover between the lung and the liver in this presentation, so we've had a nice little crash course already. I'm going to be talking mostly about rejection and a little bit about injury states in liver transplant biopsies. So like Interlung, the Interliver study is also a multi-center prospective trial. We use standard of care transplant biopsies, and it's all registered with clinicaltrials.gov. And our research within the Interliver study to date has really been in 4 major phases. So that's our initial assessment of rejection, an initial assessment of injury. We've revisited rejection since. And then at the end, I'll just talk about some of our upcoming research directions and questions that we have that we're exploring right now. And again, to reiterate what Phil said at the beginning, but a special thanks definitely to all of our Interliver collaborators. Our most recent 765 biopsy cohort represents 55 different investigators at I think 14 centers, so it's really a huge group effort. So liver transplant rejection, mainly T-cell mediated rejection or TCMR, can be diagnostically challenging by histology with low to moderate agreement between observers, especially when there's other liver diseases present. And then ABMR remains a more controversial phenotype in liver transplantation. DSA testing is not done standard at many major centers, and ABMR itself is not regularly assigned as a diagnosis, so this creates a bit of a challenge. Our objectives in this project are to increase the precision and accuracy of diagnoses in liver transplants, and also to study the underlying disease mechanisms in those different phenotypes. So for this first set of analyses, we used 235 biopsies. These were all prospectively collected from 10 participating international centers at the time. And this really represents our first look at rejection. And in this case, we used previously developed rejection-associated transcripts, or RATs, and transcript sets to analyze and interpret the molecular data. So the RATs themselves are a transcript set made of the top 200 transcripts, each associated with ABMR, and I'm showing those in blue here, TCMR in red, and then all rejection in kidneys and hearts, the all rejections in green. The RATs obviously have some overlap. There's a lot of sharing between these processes. And so if we're looking for unique transcripts, we get 417 unique transcripts altogether. Now we're going to compare these to some of our previous experience here. If we plot all the RATs, and that's what you're looking at here in these plots on the right-hand side, we see separation of the RATs revealing distinct ABMR and TCMR phenotypes in kidney, and that's the biopsy set in which they were developed. But we also see the same separation in heart. So we're basically looking at separation of dark blue and cyan dots from the red and orange, and you can see that very clearly in panels B and D, so kidneys and hearts. But when we do the same thing in liver biopsies, we do not see the same separation. That's in panel F over here. And so this suggests to us really that there's no distinct ABMR phenotype similar to that in kidney and hearts present in this population. The fact that we could not see 2 distinct phenotypes suggests that there was only one rejection phenotype at least dominant in livers, and that's TCMR. So from there, we went into our first archetypal analysis of liver transplant rejection. You've seen stuff like this just previously that looked a lot similar in lung. And so here we're looking at 4 different groups. The first one in gray, we're calling relatively normal. In red, we're looking at TCMR. In blue, injury, and this small little orange group that we were calling late. So some features of those groups are R1 relatively normal group. We found 129 biopsies belonging to that cluster with very low expression of all rejection and injury transcript sets, and also a fairly long median time of biopsy post-transplant, over 2,000 days. Now, when we look at the TCMR group, there were 37 biopsies there, all with high expression of TCMR and rejection-associated transcript sets, and a much shorter median time of biopsy post-transplant, 214 days, so within the first year. In the R3 injury group, there were 61 biopsies. Those had increased expression of tissue injury-associated transcript sets and a very short median time of biopsy post-transplant, only 99 days. So it's likely that some of this reflects injury from donation implantation. And then finally, in this late group, we're seeing high expression of injury-associated and atrophy fibrosis-associated transcript sets and a really long median time of biopsy post-transplant, over 3,000 days. When we look at the time course of those rejection archetypes, essentially just over days of biopsy post-transplant, that's what we're looking at on the x-axis here, we see something that's pretty unique compared to a lot of the other transplant populations that we look at. The TCMR phenotype especially decreases in prevalence over time. That's very unusual. In kidneys and hearts, we see it increasing at least to a peak and then plateauing or dropping off. The injury phenotype also decreased pretty steadily over time. And then in contrast to that, we see this normal phenotype increasing pretty sharply and steadily over time post-transplant, and then a more gradual increase of this late phenotype. So basically, these biopsies are becoming more and more normal as you move further out post-transplant. When we looked at some of the laboratory data for this population, we see that the normal group had basically the best biochemistry. This is what we would expect. So high bilirubin, low everything else. The TCMR group had decreased albumin compared to the normal group, increased bilirubin, increased AST, ALT, and ALP, with the ALT being the highest of the 4 groups. And then in that injury group, again, another decrease in albumin, increase in everything else, and the highest AST. Finally, that 4th group, again, only 8 biopsies, but the lowest albumin, highest bilirubin, and highest ALP of any of the other clusters that we looked at. We also took a brief look at agreement between molecular and histologic TCMR, and here we're defining histologic rejection as portal, venous, and bile duct inflammation, some greater than 1, and then looking at basically our interliver molecular sign outs as the columns there. And so we are finding moderate agreement. There's obviously considerable disagreement. This is similar to what we see in other transplant populations like heart. So our conclusions from that first attempt at looking at rejection was that early acute molecular rejection, or TCMR, is common in liver transplants, but it does become rare over time. And this is compatible with the tolerogenic properties of the liver. We also didn't find a distinct ABMR phenotype, even though we were checking for one. Couldn't find it in this initial analysis. There's a lot of different reasons why that could be. ABMR in liver transplants could be very different from that seen in kidney and hearts. It could be very rare, or it could simply just not be present in liver transplantation at all. And then our last point was that biopsies called molecular TCMR had moderate agreement, but with considerable disagreement with the standard of care assessment, and that was similar to what we'd seen in heart transplants previously. So we'll move into phase 2, and I'll be a little bit quicker in this section, but we're just going to take a quick look at what we've seen in injury. So like in rejection, there's still a lot of room for improvement in our precision and accuracy for various injury phenotypes in liver transplants, and that includes things like steatohepatitis and fibrosis. And so with this in mind, we expanded our analyses to include some molecular injury phenotypes, and we'll be using the supervised and unsupervised analyses that Pat talked about earlier. Our supervised analyses here were classifiers. and using gene expression from 337 liver transplant biopsies. So we wanted to get a good scope on the injury in our population. So we picked a diverse set of molecular transcripts so that we get a good picture at what's going on. We have 4 transcripts here that are increased after recent injury, 2 that are associated with macrophage infiltration, and then a final transcript that's associated with atrophy fibrosis or increased in atrophy fibrosis. So this is our PCA, or principal component analysis factor map, just showing us the directions in which those transcripts are traveling in principal component space. And this allows us to really interpret what the principal components mean. So principal component 1 is separating something from something else. Same with principal component 2 and 3. So if we look at these plots, we can see that all the transcripts are moving to the right. So PC1 is essentially injury versus no injury. For PC2, we can see that the transcripts associated with atrophy fibrosis are moving opposite from a lot of the transcripts that are associated with recent injury. So PC2 then is fibrosis versus early injury. PC3 is a little bit more difficult to interpret, but it looks like, because we have our macrophage transcripts traveling towards positive PC3 versus all the rest of our transcripts, that this is probably some form of inflamed injury versus non-inflamed injury. We looked at those relationships between liver injury principal components and time. We see that PC1, which was injury versus no injury, and PC3, inflamed versus non-inflamed injury, both decreased over time. And that's really telling us that biopsies get more normal and have less inflammation as time post-transplant increases. PC2, on the other hand, which was fibrosis versus early injury, increased. And this represents the accumulation of fibrosis phenotypes in the population over time. We also looked at relationships with biochemistry and found essentially as the PC1 score increases, your biopsies are becoming more biochemically abnormal. So your albumin is dropping, everything else is rising. And then PC2 and PC3 both reflect some form of abnormality at either end. So for expected, they had biochemical abnormalities at both extreme ends and then relatively normal biopsies at around 0. We also looked at developing classifiers. So this is moving back to our supervised analysis. We developed an ensemble machine learning classifier for predicting histologic steatohepatitis, and this was trained on steatohepatitis grades. And we found that this was able to predict steatohepatitis with a 10-fold cross-validated AUC of 0.83. And we moved from there into developing a similar classifier for fibrosis, again, trained on histologic fibrosis grades. This classifier predicted fibrosis with a 10-fold cross-validated AUC of 0.74, but we also found that the PC2 score was quite valuable in predicting fibrosis with an AUC of 0.8, so it gave us 2 different options. We took the top transcripts associated with histologic steatohepatitis and ran those through a pathway analysis just to see what the overall relationships were. We found a lot of terms that were associated with metabolism. And we did the same thing with the transcripts associated with histologic fibrosis and found a lot of matrix terms, so terms associated with collagens, membranes, restructuring, reorganization. So from injury, we've learned that principal component analysis using those injury-induced gene sets separated injured from relatively normal biopsies in PC1, early injury from fibrosis in PC2, and then inflamed injury from non-inflamed injury in PC3. We found a machine learning classifier trained on those histologic steatohepatitis grades, was able to predict steatohepatitis with an AUC of 0.83, and the top genes expressed in that steatohepatitis state were associated with pathways related to metabolism. And then finally, a machine learning classifier trained on histologic fibrosis was able to predict with an AUC of 0.74, and then the PC2 score was able to predict fibrosis with AUC 0.8. Top genes from a molecular fibrosis state were associated with pathways and related to reorganization, collagen complexes, and cellular structure. So from there, we went into our most recent exploration of rejection in liver transplants, and this will follow much of the playbook from the lung slides that we just saw previously. So now we're looking at 765 liver transplant biopsies. We'll be using unsupervised archetypal and principal component analyses using the same optimized rejection selective transcript sets as input. So we're just looking at separating ABMR-prob, TCMR-prob scores in PC2 like what Pat had described. And this was all validated in a training set and test set before we did our final results. So I'll skip over this a little bit. It mostly follows what we did in lung. And essentially, we found optimized archetypal analysis defining 4 rejection-related groups. So that's no rejection with 416 biopsies, TCMR with 125, a similar NKRL state with 98 biopsies, and finally an injury-induced inflammation state with 126 biopsies. The no rejection and injury-induced inflammation separate from TCMR and NKRL along that PC1. So going in this direction. The TCMR and NKRL phenotypes separate along rejection PC2 from each other, and PC3 separates no rejection and TCMR from NKRL and injury-induced inflammation. We looked at the top transcripts associated with TCMR and NKRL, just trying to suss out what's going on in each of these groups. The transcripts most strongly associated with the TCMR archetype were all annotated as T-cell and TCMR transcripts, as expected. And then the top transcripts associated with the NKRL score were all expressed in NK cells. We looked at the relationships with biochemistry as well and found that the TCMR and injury-induced inflammation archetype scores correlated significantly with abnormal biochemistry, so decreased albumin, increased bilirubin, ALP, ALT, and AST. And in contrast, NKRL and no rejection archetype scores correlated with relatively normal biochemistry. We studied the injury phenotypes or archetype groups within those NKRL and TCMR archetype groups and found that most TCMR biopsies were categorized as late fibrosis or some form of injury, and they had significant parenchymal injury and molecular fibrosis. And again, in contrast, NKRL biopsies were mainly normal by injury archetypes. They didn't have an excess of injury or fibrosis. Those NKRL biopsies compared to normal had slight increases in injury or fibrosis-associated scores like IRATS or the fibrosis classifier. And the TCMR biopsies had substantial increases in injury or fibrosis-associated scores. Actually, in most cases, TCMR biopsies had the highest mean injury-associated molecular scores of any of the archetype groups. The final thing that we looked at was associations with graft survival. Now, I will leave the caveat here that this is limited data. We have only 9 grafts that failed. However, these failures were mostly related to injury-associated molecular scores, and I've sort of put a box around them here. We're focusing mostly on those 2 green lines on this random forest. So the normal archetype score, which is absence of injury in this case, no rejection archetype score, and a late fibrosis archetype score. And we have found similar results in other transplanted organ populations. That is, injury features are generally most predictive for graft survival. So our conclusions from this latest attempt, comparing rejection estimates to parenchymal injury estimates in liver transplants reveals that TCMR does damage the parenchyma, similar to findings in kidney and heart transplants. And that's really inconsistent with this previous belief that liver TCMR causes relatively little damage. Biopsies with normal parenchyma become common with time. That's been consistent through all of our different analyses in liver. And finally, that optimized rejection algorithms indicate that some liver transplants do manifest this NKRL state that is well tolerated, at least in the short term, and enriched in NK cell transcripts with relatively little parenchymal injury or biochemical abnormality. But we want to keep looking at that more. So I have some extra slides at the end if there's questions, but I think that basically caps it off. So thank you very much for your attention. We can move into questions, Phil, if you'd like.
Okay, do you want to move out of your screen share?
Yeah, definitely.
Sorry, we've gone on a little bit longer than we intended, but well, I think our main points today are, number one, we've seen the central feature of T-cell mediated rejection, and that opens some therapeutic opportunities that we've never seen before. We could get the quarterback. Second, in lung transplants, we see that the mucosal biopsy gives us remarkable information. It correlates with what we see in the transbronchial biopsy, but it's much safer. And in fact, some of the information is actually better than what we see in the transbronchial biopsy. And remember, the Achilles heel, the lung transplant, is the airway. And the third thing is that that in the liver, we are seeing T-cell mediated rejection. And in all of these organs, we see consistent things in between heart and kidney. But in lungs, we're not seeing an antibody-mediated rejection phenotype. Nothing convincing as a disease state, although there may be some activated NK cell states, which are probably relatively well tolerated and don't need treatment. Anyway, that's what I think we're seeing. Thanks. Questions?
I have a couple of questions. One is on the liver side. What is no rejection? I didn't understand what that was.
It's no rejection.
Yeah, just
no
the no
rejection
phenotype. Yeah, so low.
But it's still associated with graft loss.
So the no rejection, when we're looking at random forests, you have to keep in mind that the bars don't indicate the direction. So it just says it's significantly associated with graft failure. So the absence of something can also be important for not losing your graft, or the presence. Random forests never indicate the direction. Yeah.
So the no rejection state should be seen as normal. So we can sort of combine the no injury and no rejection and see biopsies which are normal and biopsies which are injured. But the T-cell mediated rejection state is a bad state to have because it leads to parenchymal injury, and the parenchymal injury in all the organs is what really drives outcomes.
But I guess what I'm seeing here, what we're seeing here, I'm guessing, is that a lack of normal phenotype is what's associated with rejection. Okay, that makes sense.
Well, no, T-cell mediated rejection is a very characteristic state in each of these organs. It's absolutely like a fingerprint, and these all 22 cells are changing virtually the same in any of the organs when you get T-cell mediated rejection. In the lungs and the hearts, there are some states which seem to be enriched in NK cells, but they're not activated, and they don't seem to be associated with the state. And in fact, on average, they seem to do a little bit better than the other grafts. So we cannot say that there's any disease state in these organs which is like the disease state which so clearly emerges in hearts and lungs. One possibility may be that the effector mechanisms for antibody binding, either the density of antigen or the amount of antibody you can produce, or more probably the amount of NK cells in the circulation which are capable of this, are simply finite. And with an extremely large microcirculation, you just can't reach critical mass. You can in hearts, you can in kidneys, because they're about 1/10 the size of the microcirculation, but you can't in lungs and livers.
Interesting idea.
That's a formulation of what we're seeing. We're all very interested in the other ways that NK cells could see allogeneic tissue, the missing self, and there are so many ways that the NK cells can interact with tissue. But throughout the history of organ transplants, we've never really seen a phenotype which is independent of antibody. It doesn't mean we haven't missed it, but we could have missed it. But I think we probably think right now there are only two major disease phenotypes, and in lungs and livers, there's only one rejection phenotype based on allorecognition.
So my second question is, in lung, if you look at biopsies that are histologically called as ABMR, what do you see? Is PC1 high, or is just sort of like
nothing
random?
First of all, they're very rare. Secondly, the pathologist and the clinician don't agree. And thirdly, we just don't have enough to make any strong statements. But I think that the formulation of antibody-mediated rejection in lungs is probably driven by the idea that DSA associated phenotypes will be bad. And we're just not seeing any molecular counterpart to that.
Okay.
Well, this is fascinating, and the fact that there seems to be a disconnect between what people are calling AMR or ABMR, in clinical lung transplant and the gene arrays that you're seeing, it's really intriguing. And, although I realize that people are not rodents, but at least in the rodent models, the murine and the rat model of lung transplant, especially with major MHC differences, you do see, in my understanding, AMR for the transplanted lung. What I'm kind of wondering is that, and we also know that the phenotype of CLAD, which is the restrictive allograft syndrome, at least clinically, it seems that most of those are DSA positives, and those are suspected, the restrictive CLAD, to be related to AMR, as opposed to obstructive CLAD. So, there seems to be a little difference in what we're seeing clinically. But what I'm wondering is that histologically and radiographically, when we see these very bad cases of AMR in lung, the histologic correlate is alveolar damage. It's diffuse alveolar damage. It's injury. And so I think you're quite right about that. And the fact that maybe the cellular component is different in the cases of, that you're looking at, AMR, are not the severe cases that are presenting clinically, where you see alveolar damage, and the cellular component of it is a very neutrophilic rich infiltrate. So it's a neutrophilic, at least in my understanding, a neutrophilic capillaritis and diffuse alveolar damage as really the manifestations that we see. So maybe it's just we don't have enough of these cases that are in your clinical set.
Well, I'm going back and forth with Kieran about this. One of the situations we see in all organ transplants is the non-adherent under-immunosuppressed situation. And that starts off with a T-cell mediated rejection process, which is often very devastating. And then with time, as in COVID, the T-cell response morphs into the antibody response. So what you'll see is these cases start off as non-adherence T-cell mediated rejection, and then DSA appears. And in the case of kidney and hearts, the DSA can be the long-term problem after you've treated TCMR. But in fact, you're often left with devastating parenchymal damage. So I think that the belief systems that have generated the formulation of ABMR in lungs and in livers are probably incorrect. They start off with the belief that if DSA is present, there's a high probability that that's ABMR. It's not, because a lot of the time, the cognate T-cell response will be followed by a DSA response. So the non-adherent patient will often be showing DSA as well as severe TCMR. And I think that they've just created a very serious disease state, but the disease state is probably due to non-adherent TCMR followed by DSA. And I think that the key is don't miss the TCMR, because if you can save that person's life, it's by suppressing the TCMR. And I would say try to sack the quarterback. And listen, there's a very close relationship between TCMR and CLAD in the molecular diagnoses. And that's particularly in the 3BMBs. And I'm not sure that we're not seeing smoldering TCMR much more frequently. This is the TCMR2. Now in kidneys and hearts, there's a TCMR state which is associated with fibrosis. And that's seen late. The TCMR is less intense, but the fibrosis and parenchymal damage is much worse, and it's a bad state. And I think that may be the beginning of the CLAD state. In at least some cases, the TCMR anticipates that it will be producing CLAD. We can't see it all the time, but there's certainly an association between CLAD, and it's particularly clear in the mucosal biopsies.
And that would fit very nicely with some of the data that was published by Alan Glanville many years ago, that even these cases in lung of grade A1 rejection, where many centers don't even treat that, but if you've had more episodes of A1, despite their allegedly benign nature, that there's a higher risk of CLAD.
Alan Glanville was right about a lot of things. He's one of the many figures we miss in our field.
I echo that completely. I miss him dearly.
Well, Giovanni, any comments? Or Kieran wasn't able to join us today.
Yeah, no, sorry. I had another meeting earlier on, so I was only about 30 minutes. I came in about 30 minutes later. I'm going to have to review the recording for the lungs section.
Okay. Well, listen, I know many of you have full lives, so I'll thank you very much for joining us, and thanks very much to Pat and to Katelynn for taking us through this work. We have 10 billion data points in what we've just shown you. 10 to the 7th. Anyway, so it's interesting to see how that can be formulated into clinical states and perhaps guide clinical trials and guide treatment, which is the next phase. This is the new era in transplantation, I think. And we're going to be questioning everything and getting some things wrong too. Okay, thanks very much. Over and out.
Thank you.
Thank you, everyone. It was beautiful.
Thanks, everybody.